Alright, so now we're moving on to chapter 2 where we're going to talk about the cell and some of the things that go into the cell, so different components, and how cells can organize themselves and function on a very basic level. So we're going to cover a few things within this chapter. We're going to talk about the structure and function of the cell membrane, including talking about how things kind of come in and out of the cell. We're going to talk about the different organelles and what they do, and then the structure and contents of the nucleus. And then we're going to talk a little bit about how cells go through their daily lives. How do we make one cell into two cells or more? And then also a little bit about how cells differentiate and become more specialized. Because one thing we're going to talk about in the relative near future is about the different types of cells and how they look and the functions that they do, at least to some degree. And so we have to understand how a cell gets to that point in the first place. So let's start off with the cell membrane. So there's a few things we want to accomplish by the end of this, and that is we want to talk about the components that make up the cell membrane, some of the major features and properties of it, and then how things can get in and out, and how different materials have a different experience with the cell membrane, and then talk about some of the different ways that things can come in and out of these cells through that membrane, through both passive and active transport, and talk about some examples for each of those. So let's talk about the basic structure of the cell membrane first. So there's really a few main components. We have the phospholipid bilayer, which is a double layer of phospholipids, and then we also have cholesterol in there, and various proteins, glycoproteins, things of that nature that are embedded on and in the membrane itself. And the whole point of having a membrane, right, is to create a barrier, right, that divides the inside from the outside of the cell. So it divides the inside of the cell from the external environment. And then also, it's not this just wall. It is dynamic, it's fluid, and there's a level of control and regulation in terms of what materials can get in and what materials can get out. And it's very controlled, right? It's very regulated. It's not just anything can get through or nothing can get through. Certain components, certain materials will be able to get in and out very easily. Other things may need a little bit of assistance, and then some things are just going to really need help getting through that membrane if they can at all. So let's talk about the phospholipids first. Now the phospholipid is an interesting molecule because it is both hydrophilic and hydrophobic at the same time, meaning water-loving and water-hating or water-fearing. So we have like this duality to the phospholipid. When we've learned about lipids in the past, typically you learn that a lipid is hydrophobic, that lipids do not like water, they don't mix well with water. The phospholipid is a little bit different because we do have a side of it that still hates water. It does not mix with water, and that's the hydrophobic tails. But we also have this hydrophilic head with a charged phosphate on it, which is why it's hydrophilic. And it's this duality of the phospholipid is going to create a membrane that gives us control based on things like size, because if you're too big to pass between these phospholipids, it's going to be difficult, but also based on charge. And so looking at that, we have some control. And this is going to come into play not just in this chapter, but we're going to see this come back later, especially when you get into physiology. So when we look at the basic structure of the phospholipid, right, everything here in red, right, this red circle is the hydrophilic side, right, and this is the side that would prefer water. And then these two little lines down here are the hydrophobic tails. These are the parts that do not like water. And so when we see the phospholipids all together, they will spontaneously form this bilayer. And when we say spontaneously, it means just naturally, without real any input. They will organize themselves in this way. And the reason they're organized this way, right, if we look at this diagram, we have the outside of the cell, also known as the extracellular space, and the inside of the cell, or the intracellular space. And both the inside and the outside of the cell are going to have a lot of water. And so that means that the phosphate heads, right, that are hydrophilic, they're going to be attracted to the water. So these phospholipids are attracted to the water on the outside. These phospholipid heads are attracted to the water on the inside. And that creates this kind of sandwiching of the hydrophobic tails because they don't want to be near the water. So they're going to push themselves on the inside of this layer where they feel protected, right? They're kind of sandwiched and squished between the two hydrophilic head sides that are facing the water, creating this lipid-based hydrophobic middle layer. So we think like a sandwich, right? The heads are like pieces of bread, and then the tails are like all the fixings that are on the inside there. Right? And the fixings don't want to touch the plate, right? They don't want to touch the environment. You want the bread on both sides to provide that protection there. Right, so we have our phospholipids forming this bilayer that creates this this barrier that is hydrophobic, right, in that middle there. But the phospholipids are not the only things there. That's not enough. That doesn't provide enough control. So we have some other things that are embedded in there as well. We have membrane proteins. Membrane proteins are can be in two different forms based on how they're embedded in the membrane. We can have integral proteins. These guys are actually embedded into the membrane, and peripheral proteins, which are on the surface, right, either on the outer surface or the inner surface. Integral proteins, the ones that are embedded, this can include things like channel proteins, which allow things to pass through from one side of the membrane to the other, or cell recognition proteins, so things that are going to help the cell recognize things around it. And both of these can cause an effect within the cell. So let's take a peek, and we'll talk about some other things as well while we're looking at this figure. But here's that peripheral protein right in the membrane there, and you see how it just kind of is embedded on one side. It doesn't pass all the way through. The integral membrane proteins, you can actually see it from both the inside and the outside. So our channel protein is a good example of an integral membrane protein because you can see that we created like a pore or an opening all the way through, right? And we'll talk about channel proteins a little bit more in just a bit. We also have things like glycolipids. So this is like a protein, this is like a lipid and a carbohydrate together. We also have glycoproteins, which is a carb and a protein linked together. We can use these for things like recognition and signaling and whatnot. And then we also have cholesterol. We haven't mentioned this yet, but cholesterol is a lipid, and this is going to help give some stability to the membrane. It's important to note that this membrane here is fluid, right? It is not a static membrane that just stays still. It's not a brick wall. These phospholipids and all these proteins can actually move around. So it's kind of like if you had a very dense crowd, people can move within the crowd, even though as an entire crowd it looks very solid, right? You're not going to easily get through the crowd, right? It's going to take a lot of work, but the crowd is not stiff, right? The crowd is moving. So we can kind of think of it like this is a really densely packed crowd that is creating this barrier between the inside and outside of the cell. Cholesterol provides some stability because the fluidity of this membrane can change based on things like temperature. Cholesterol gives some stability to that so that we don't have a constant changing or constant flux of the fluidity of the membrane. Now, plasma membranes don't have to necessarily be just totally flat. We can have different extensions of the plasma membrane. Some of these we will see in certain cells and certain tissues throughout the course, so we want to be familiar with these. We have microvilli. These are little folds to increase surface area. So instead of being nice and smooth, we have little extensions that come up and down. And as we do that, we're keeping the volume the same, but the surface area, the outer area of what we're looking at is getting higher, right? So the surface area is way higher compared to the volume. And this is really good in areas where we're doing something like absorption, because the more surface area you have, the more absorbing you can do. So we'll see things like microvilli in things like the digestive system where absorption is very important. We also have cilia. Cilia are going to be little extensions that are almost like little hairs, right? Like little fingers that produce movement. And so if we need to move something along, cilia can be very helpful. We'll see this in places like the trachea where we have mucus and debris that needs to be moved, and so cilia in the trachea can help move that mucus, move those things along. And then flagella, this is similar to the cilia in that it does produce movement, but our structure is different. Whereas the cilia are short and numersome, right, or in high numbers, the flagella are long tails, and you only have one or two of them usually. So like for example, we look at sperm. Sperm has a flagellum, a single tail-like structure, a single flagella, to help the sperm swim. So here's just some of those examples. So we have like the microvilli here. You see those little projections up and down, up and down. This is going to increase how much surface area we have in contact with the outer space, right, compared to the volume. So if we need to absorb a lot, that helps a lot. Cilia are going to be these longer extensions used for movement, so these cilia can actually move. So usually you see them moving together like in unison. And then here's our flagellum in our sperm, where it's this one single long extension that functions like a tail, right, to help swim. So how do we get things from one side of the membrane to the other? Because again, we have to let some things through. The cell has to interact with its environment in some form or fashion. So let's go into how we move things across the membrane. It's important to note the membranes are what we call selectively permeable, right? That means some things will be able to get through the membrane easier than others, right? Some things will get through easier than others. Some things can get through with no effort at all. Other things may need a little help through like a channel. And then some things are going to need actual active work to bring them in or out. So not everything has the same pathway to get through the membrane. And so we're going to talk about two modes of transport, and we'll break them down into smaller chunks as well. But we're going to talk about the main two modes, which is passive and active transport. Passive transport is called passive transport in that it doesn't require energy for this to occur. It's just going to happen without any energy input from the cell. And then active transport is going to use energy. And usually the difference between passive and active transport has to do with how we're moving from one concentration to another, how we're moving along the concentration gradient. Because concentrations of solutes or concentrations of these components that we're trying to move, the concentration is not the same on each side. One side might have a high concentration, the other side may have a low concentration. Passive transport tends to move from a high concentration to a low concentration. This is the equivalent of going with the flow, right? Flowing downstream. So if you are going tubing right down the river, you're going with the flow. You don't have to put any active effort into moving down the river because you're just going with the way the water is flowing. Same thing with passive transport in that we're moving from a high to a low concentration, which is the natural expectation, the natural thing that will occur. Active transport, we're going from a low concentration to a high concentration. So we're actively going against the current. So this would be the equivalent of trying to swim upstream, right? Swim against the current of the river. So if you've ever done that, you've noticed like you have to really put some effort in, right? You have to actively swim and burn energy to do that. So passive transport is free. You're going with the flow until equilibrium is reached, right? Until both sides are equal. Whereas active transport, you're actively trying to make the concentration gradients stay or get more different from each other. So like I said, passive transport, we do not need to use energy for this to happen, so we're moving down the concentration gradient. We're going with the flow from high to low. This is also known as diffusion, and there's a few different breakdowns within diffusion. We have simple diffusion, facilitated diffusion, osmosis, and filtration. So let's talk about simple diffusion first. So simple diffusion, in this case, we have these small uncharged molecules, and we have our membrane bilayer, right, our plasma membrane. And on one side, you see that our molecules are highly concentrated on this side, but on this side, they're not even there, so the concentration is 0. And as time goes on, these molecules will make their way to the other side where there's very little of that molecule. And we're going to do that until both sides are equal. Once we're equal, this molecule might move across, but for every molecule it moves one way, another molecule is going to go the other way. And so we have a net zero movement in this case, right? So the whole point is we're trying to reach equilibrium. These molecules can push their way past the plasma membrane. They can push their way past the phospholipids to get from one side to the other until each side is equal, right? So if you've ever taken like a drop of food dye and put that drop in a beaker of water or a cup of water, you'll notice that if you give it time, that drop of dye actually disperses throughout all of the water, right? And that's just random movement of the dye molecules until they're all kind of equally spaced from each other. This is kind of the same thing. The only difference is we've introduced a membrane in between. Now, the reason these molecules in this case are able to get through, because I know you're thinking, well, what's the point of the membrane then if they can just get past? The reason these components, these molecules can get through is because they're so small that they can push their way through, and they do not have a charge. So because they don't have a charge, this middle hydrophobic layer is not going to repel them. Right? And those are the two things. You cannot be large, you cannot have a charge to follow simple diffusion. If you are a large molecule or if you have some sort of charge, you're going to need a different pathway to get across the plasma membrane. And that's where facilitated diffusion comes in. So let's say we have something that is too big or something that does have a charge. We can still use passive transport to get them from one side to another. We just need basically a pore or a doorway to get through. They can't push their way through the phospholipids, but if they are given a channel protein, they can easily pass through. Right, so a channel protein or a carrier protein, both of these have a similar concept in that they're allowing something to move from a high concentration side to a low concentration side. Right, the difference is we can only come through this particular point rather than push our way past the phospholipids. But the concept is the same. We're still moving from a high to a low concentration. That part is still the same. The only difference between simple and facilitated diffusion is whether or not we're using a channel protein. And then osmosis, we're looking at water's movement in particular. And the reason we make a difference here, where we differentiate osmosis from diffusion, is we're looking at which component is moving. When we were looking at simple diffusion, we were focused on the solutes, right? The things that are dissolved in the water moving across. In osmosis, we're not talking about the solutes moving, we're talking about water moving. And so it kind of feels a little opposite in this aspect compared to the simple diffusion. So remember, simple diffusion, the solutes were moving from a high to low concentration. In osmosis, the solutes cannot move, right? The solutes are locked. And so let's say we have this beaker and we have a membrane in between dividing the beaker in half. And we have a high solute concentration on the right here and a low solute concentration on the left. The solutes are locked. They cannot pass through the membrane. But water can. And so in this case, water is going to move through the membrane to dilute the high solute concentration. This is to equalize the solute concentrations on both sides. Since the solute cannot move, the only way to equalize their concentrations is for water to move to the higher solute side to dilute it down. Right? So we would see in this case, water would leave the left, move over to the right, until enough water was on the right that the concentrations for left and right solutes are the same. So this water level on the left has dropped, right? Concentrating the solutes on the left. Water has moved over to the right, diluting the solute concentration on the right. And so we can describe these solution or solute concentrations with a few different terms, right? This osmosis is happening because the solutes are not equal on both sides, and they can't necessarily become equal on both sides of the membrane. So when we're talking about the solutes or the solution concentrations, keep in mind we're talking about the environment that our cell is in. We're talking about the solute concentration of the outside of the cell. But let's start with the easy one first, which is isotonic. In isotonic solutions, the concentration of solutes is equal on both sides of the membrane. So the inside and the outside of the cell, solute concentrations are equal, which means a net zero movement of water. That means, it doesn't mean water's not moving at all, it just means that for every water molecule that leaves the cell, another one is coming in, right? So if someone leaves and someone else comes in, yeah, the individual molecules are different, but the overall concentration of water is the same. Now, if the outside of the cell has a lot of solutes in comparison to the inside, we call that solution a hypertonic solution. So the concentration of solutes inside the cell is low, the concentration on the outside of the cell is high. This is going to draw water out. It's going to pull water out of the cell, dehydrating it, right? Because the solute concentration is so high on the outside, water gets pulled out towards that high solute concentration. And then hypotonic is the opposite. The high solute concentration is inside the cell. The outside of the cell has a low concentration of solutes, which means water moves in to dilute the solutes inside the cell. With hypertonic solutions, what we see with animal cells is that water gets pulled out and we experience what's called crenation, right? This is a shriveling of the cell. Think like going from a grape to a raisin. That would be what would happen in a hypertonic solution. Your cell, if you think of the grape like your cell, you put it in hypertonic solutions, it's going to draw that water out. Your grape is going to become like a raisin. With the hypotonic solution, this is like overflowing a water balloon, right? Where you're pumping so much water in that it gets really swollen, really tight. Now, with animal cells, we don't have any protection from too much water coming in. So that means if too much water comes in, we can actually experience not just a swelling, but we can experience what's called lysis, which is the bursting open of the cell, right? We've put so much pressure, so much like water pressure on the membrane that it literally gives way. And it's like if you overfilled a water balloon, that water balloon would pop. So in a hypotonic solution, that's what we would see happen to an animal cell. So here's our little picture, right? So isotonic, animal cells love it, right? Solute concentrations about equal. Water moves in, but also water moves out. It's a net zero, right? So here's our little red blood cells, and they're nice and healthy and happy looking. In a hypertonic solution, right, there's a lot of solutes in this black background you see, and so it's pulling water out of these cells. And so like a grape turning into a raisin, right, we see that crenation, that wrinkling and shriveling of our cells. And then on the hypotonic side, all the way on the right, we see that they look way more puffy, right, overfilled, because water is flooding in because the high solute concentration is on the inside of the cells, and the outside of the cell, the black background, has very little solute in it. And we see that this one cell here has actually experienced it to the max, where the membrane has actually given way because so much water has flooded in, the membrane cannot handle the pressure and it bursts open like a balloon that was overinflated. So those are the ways that we can pass through passively. It does not require any sort of energy. But active transport, this is where we're going against the concentration gradient. We're swimming upstream, and so this does require that the cell puts energy into this effort. We're going from a low concentration to a high concentration. So rather than trying to reach equilibrium where both sides of the membrane are equal, we're actively trying to keep the concentrations different on each side of the membrane, and that takes effort. There's a few different types. There are different types of protein pumps. We also have endocytosis and exocytosis. Endo and exocytosis are forms of bulk transport where we're moving lots of stuff all together. So we're going to talk about the most common pump, and that is the sodium-potassium pump. This is one of the most common forms of active transport in the cell, the one that you hear about the most, basically. And so what we see here is that there's a lot of sodium on the outside of the cell, and there's a lot of potassium on the inside, right? So sodium is shown with these orange hexagons, and potassium is shown with the yellow ovals. And so what we see is, even though the high concentration of sodium is on the outside, the cell is actively pushing sodium out, right? So we're trying to eliminate or keep the sodium level really low inside the cell by pushing it out and out and out and out and out. And we see the opposite happening with the potassium, right? There's very little potassium on the outside of the cell, and yet we're finding all the potassium we can on the outside and pumping it in, keeping that potassium level really, really high on the inside. And so we're forcing it to have this difference in the concentrations, where we're forcing there to be a high sodium concentration outside, and we're forcing it to have a high potassium concentration on the inside. And this requires ATP, right? And it requires it because we're literally going to cause a physical change in shape to these membrane proteins, right? To these carrier proteins. We're going to cause a physical conformation change, a physical change in shape to link those sodiums in and then dump them out, and then bring potassium in and dump them in. The big thing you need to remember, though, is the use of ATP. That's one of the biggest things. And you'll talk more about sodium potassium pumps in physiology, because these are involved in some very important physiological reactions. Endocytosis, this is where we're bringing something in. So endo, we think in. This is so that we can bring in maybe like a large particle, bring in fluid with solutes dissolved in it, or to bring in very specific things using receptors. And so there's three different forms of endocytosis. We have phagocytosis, think to eat, right? So phage, we think to eat. So this is like something our white blood cells can use to bring in like a large bacterial cell. We actually create like a little pocket of the plasma membrane and then pinch that pocket of membrane off to form what we call a vacuole or a vesicle, right? And then pinocytosis, we're again forming little pockets of the membrane filled with the fluid as well as dissolved solutes. And then we pinch that off and we create a vesicle that has the exterior fluid with the solutes dissolved in it. And then receptor-mediated endocytosis, this is kind of similar to the pinocytosis, except now we want specific solutes brought in. And so we have membrane receptors that are embedded on the outside, and that these particular solutes, these particular components can actually bind with the receptor. And it's very specific. This is called a ligand. When it's something specific, we're trying to bind to a receptor. And so they fit together kind of like a lock and key. You know, not every key can be used on any lock. It's very specific. And so the ligand is specific to the receptor. So if we only want to bring in stars, in this case, and not the squares, we can use receptors that only bind with stars to then bind a bunch of stars to the receptors, and then again form that pocket, pinch it off, and bring it on the inside of our cell. Exocytosis is the exact opposite, right? We're going to have a vesicle that has something dissolved in it, something on the inside of it that we want to dump on the outside of the cell. And so it's just the opposite. We have the vesicle, it fuses with the membrane, and then kind of like if you were to turn your pocket inside out, right, to dump the contents of your pocket on the outside, it's kind of what we're doing, right? So your vesicle is going to bind with the membrane, and then is going to push that membrane out, right, opening up that pocket, spilling the contents outside of the cell.